All right, you guys, welcome everyone to night four. Night four, you guys, amazing. I am so happy to see you guys. We are going to have a fun-filled night. I am on the Eastern time zone, so it is 7 o'clock at night where I am. So we're going to be going into the wee hours of the night. So tonight is night four of our crams. I'm hoping that some of you guys have attended the other sessions, and if not, you are able to access all the recordings. And then tomorrow we have a grand finale, so that's pretty exciting. As always, please make sure you follow us at Think Vibable on social. Tonight we are going to have several student hosts. I am Sabrina Shumate. I am a teacher in Virginia Beach, Virginia. I teach Cox High School. I teach at Cox High School, and I teach AP Biology. And I have some amazing student hosts with me. So, student hosts, if you could introduce yourselves with your name, where you're going to be presenting from, and something on your vacation bucket list. Where's somewhere that you want to go that you for sure it's on the list?
Oh, so I can go first. Hi, everyone. It's nice to see you guys once again. I'm Madison, and I am coming to you guys from Illinois. And one vacation area I would love to go to is Italy. I just think it's so beautiful and so fascinating.
Hey everyone, I'm Joyce. I'm coming from Maryland, and a vacation place on my bucket list is Greece. Yeah, I just think it's like really pretty, and there's so much like culture within the food.
Hey everyone, my name's Jeffrey. I'm from Minnesota in Wayzata, and I want to go to Glacier National Park for the third time because it's just so good. Thank you.
Perfect. All right. Thank you guys. I would say that my bucket list, it used to be Hawaii. I've always wanted to go to Hawaii and I got to go and I loved Hawaii. But next to my list is Yosemite. I've never been able to go to Yosemite and I really, really want to go. So that's on my list. So if you guys have somewhere that's on your list, feel free to share it in the chat and we can all pretend that we are going there essentially. All right, so I'm going to go ahead and pass it over to Madison, who's just going to kind of do a little introduction, and then we're going to go ahead and go right into what we're going to do tonight.
Perfect. So here's our cram schedule. We went through already 3 nights, which I'm so proud of you guys, other cram sessions. Today, we're going to do some more skills and drills, but with some calculations and graphing problems, which you guys were asking a lot yesterday. So we have it for you. And don't hesitate to come to tomorrow's meeting on our grand finale, where us student hosts are going to show you some reviews of units 1 to 8, reminders of test taking strategies, and some tips and tricks. For the agenda today, we already went through a welcome and initiatory and introductions. Throughout the night, you guys will be doing mostly science practices. However, we'll have some math practices as well. And lastly, a wrap up. And now I'm going to give it you guys to Jeffrey.
Okay, so well, if you want to actually do good for this cram and you want to actually use like the resources we supply, you might as well take notes. You'll be able to access our Cram Hub, which has basically everything. It has the slides, it has a study guide that you can use to follow along with. Use the chat, you can talk with other people, but you can also ask for help there. And usually one of us or another person will be able to answer you pretty quickly. And then for the QA tab, we usually use that like for our formal questions. So if you ask there, you probably will get an answer. So yeah. And then our community agreements, number 1, be supportive, don't be mean, be patient, and be kind. Yeah, pretty basic. And then I'm going to use this time to sell out Fiveable. So win $100 and be reimbursed for your AP tests, which are known to be super expensive, by simply making a TikTok or Instagram post. So yeah, make sure to do that.
All right. Thank you, Jeffrey. All right, you guys. So before we get started, I want to make sure that you have everything that you need. So make sure you have something to write with, something to write on, water, snacks. We will be taking little mini breaks because today is kind of a heavy math day. And so I know we just need like a moment to step away. And then make sure you have a calculator of some kind. Okay, so go ahead, get yourself ready. And I'm going to give you a warning in advance. So I have two kids. I have a 6-year-old and a 9-year-old. So a kinder and then one in 3rd grade. And we were talking about the crayons, and I was trying to come up with some fun questions to ask you guys. And I let my kids ask the questions. So I told them that I would ask their questions and then I would share some of the funny answers with them. So some of the questions that my 6-year-old, 9-year-old came up with are probably the cutest things in the entire world. So let's have a little fun tonight, and we got this, night 4, okay? All right, so first thing I want to know, where are you joining us from? Where are you at? What time zone? What state? Let's give a shout out to where you're at. Rachel, I love you. New York, lots of New York. Ooh, we got some Florida. Toronto, nice. Thailand, very awesome. I love Virginia, another Virginia. Beautiful. We're from all over. Thank you guys so much for being willing to check in and spending your night with us. Okay, our test is Wednesday. We got this. We totally got this. Love it. Lots of Virginia. All right, so I'm going to ask you guys the first question that my 6-year-old came up with. Okay, you guys ready for my 6-year-old question? I think it's the cutest thing. He wants to know, if animals could talk, which animal do you think would be the nicest? Remember, he's 6. So if an animal could talk, what animal do you guys think would be the nicest animal? He asked me that question and I was like, I don't know. There's so many. What do you guys think? A panda, a goat. You think a goat? Oh, bunnies, axolotl, yes. Dogs for sure. I agree. A pangolin. Interesting. Oh my gosh, I think my kids are gonna think this is hilarious. Oh, otter. I love otters. Oh, copy. Oh, you must have done March Mammal Madness. Very nice. A manta ray. Oh, Pipple. I love Pipples. I have Pipple. So cute. Okay, I will for sure share those answers with my son. He will think that they're really, really cute. And he came up with this really random question. Okay, so before we get started, I want to know, and Madison, if you could help me put the poll on. We have a poll. We want to know how many total AP exams will you take this entire testing season? So for the 2 weeks, including AP Bio, how many AP exams will you take? So if you could, on the poll, Miss Madison will go ahead and turn the poll on, and then let's go ahead and see how many total AP exams. So again, on the far right, you should be able to hit the poll, and then we should be able to take a quick little check-in. Oh, goodness gracious, Lordy Lou, you guys, 5 of you had to take more than 6 exams? Okay, so most of us have taken 2 or 3. Oh, goodness gracious. You guys are rock stars. This is the hard time of the year. Then it's smooth sailing after this. So 23 of you, only AP Bio is your only exam. Lucky dogs, nice. All right, so it looks like majority is taking about 2 to 3. 8 of you had to take 6 or more. You guys are rock stars. All right, you guys, thank you so much for sharing with me how many exams you had to take. We appreciate you being here. All right, so let's go ahead and dive into it. So Jeffrey, if you could go ahead and go over the formatting with everybody just to make sure that we're all on the same page.
Okay, so tonight, well, the test is going to be on Wednesday at 12 p.m. It's going to cover all 8 units, and then it's going to be 3 hours long, 3 hours of hard work. So here are the units, as you can see, and today we're just going to be going over all of them, but we're going to be using some of the more obscure math problems to go along with. Okay, and then here's what the exam looks like. So here's what you should be expecting inside the AP Bio exam. So you should be expecting 60 multiple choice questions and 6 FRQs. So yeah, and then these will cover all the following, as you can see, and we're going to be refining all your skills with a bunch of practice. Okay, and here is the science practice that we're going to be using. So we're going to be looking at data, so for example, graphs and stuff, and we're going to be testing you on how well you can analyze them. We're going to be doing statistical tests, so as you might have heard, for example, chi-squared, and then we're going to be doing argumentation. So learn how to make an argument.
Beautiful. Thank you so much, Jeffrey. All right, my loves. So before we get into the whole bunch of math, let me know, where are you at right now? Here's a little mood board. What are we feeling? Is your brain drained? Are you a 2? Are you feeling hyped? You're a 5? How are we feeling right now? What do we think? We got some 2s. Aw, love bugs. All right. Well, hopefully we don't make it worse. We just all need a good recharge, okay? We will work together. I promise. Any 6s? Anybody just utterly exhausted? We have a lot of 9s. Okay. All right, my dears. Thank you for sharing how you were feeling. We are almost there. Like literally almost there. It's the last week. Okay. AP Bio's on Wednesday. You have finals on Friday. Then you get some breaks. We have SOL testing at the end. I don't know if you guys have standardized testing as well. And then it's summertime. Okay. All right. Thank you for sharing with me how you guys are feeling. All right. So our focus tonight is going to be on FRQs. Okay. So on the actual FRQs, we are going to talk a lot about math. So that's why I asked you to make sure you had a calculator ready so we could do some practice problems. So in terms of the FRQs, I want you to know that they actually give you the format of what they're going to ask. They just don't give you the topic. Okay. So we know that we're going to have 2 long FRQs and then we're going to have 6 short FRQs. And so for the 2 long FRQs, the first one is going to be some sort of scientific scenario. It will be something that you would have never seen in your entire curriculum, okay? It'll be just this huge scenario going on. It'll be some sort of experiment, some sort of data table, some sort of graph. And then you're going to have multiple parts that will go with that graph or data. And then same thing for the second question. The only difference is for the second question, you're going to have to create the graph. So the first one comes with a graph and the second one doesn't. But again, it will be kind of this random scenario situation that happens. The difference between a long FRQ and a short FRQ is not going to be in your response to the question. It's going to be the amount of parts that the question has, okay, or the amount of points that the question has. So for the first and second, don't be freaked out when you see long FRQ and think that you're going to be writing these long essays. You're not. It's just that it's going to be worth a lot of points and you're going to be going ahead and just, you shouldn't be having to write more than a paragraph for any one of these situations, okay? So 1 and 2, long FRQ, just remember multiple parts, okay? Anything that's bolded represents a point. So one of the parts might be worth 4 points. And what I want you to know for your FRQs is that it has to be in pen. If you mess up, you do a single strike through and you do not have to do them in order, okay? So maybe you would know part C, but not part B. That's okay, just label it. So you would do C and then put it on there and then B and then put your answer. There is no intro, no conclusion, no hook, no thesis. Just say the answer. That's all you have to do for AP Bio, which is phenomenal. Maybe you know half of part A, but you don't know the other half. Just do what you know, okay? So question 1 and 2 is gonna be some sort of experiment that includes a data table and a graph. Number 2 is going to be for you to make a graph, and you should always make a graph because that's kind of like easy points right there, okay? Question 3 is going to be some sort of lab investigation. So it's going to kind of relate to a lab that you were supposed to do in your AP Bio class. And if you didn't do the lab, that's okay because your teacher covered the content of it. So just through context clues, you'll be able to kind of figure out the question that they might be asking. So here's kind of an example of a lab-based question, okay? So this deals with transformation and it deals with plasmids. Does anybody remember what a plasmid is? Can you tell me in the chat, what is a plasmid? That's kind of a big science word. What is a plasmid? Does anybody remember what's a plasmid? Beautiful. Very good. Very good. Good job, you guys. Okay. So bacteria don't have a nucleus. They kind of have a nucleoid area. And a plasmid is a little bit of DNA that's circular and independent of the main genome itself. And usually it codes for a very specific trait. Okay, so that is a plasmid. Plasmids are unique in the fact that they can be transferred from one bacteria to another, and that's called transformation. So when I think of the term transformation, I think of it being transferred. And so when that bacteria transfers its plasmid to another bacteria, that bacteria, just through protein synthesis, transcription, and translation, can go ahead and start expressing whatever that trait is going to be. And or if you have a bacteria that dies, it's going to leave its plasmids around, and another bacteria can come by and kind of uptake that. It's called assimilation, and start expressing those particular traits. But we use it in biotechnology because we can actually cut out parts of plasmids and then kind of put in other bits of DNA and create recombinant DNA. And then we can go ahead and have bacteria express certain things for us, which is kind of interesting. Okay. So here's kind of just like a little diagram. You're going to get lots and lots of diagrams and models on your FRQs and multiple choice. And what I can tell you is that almost everything you need to know is in the diagram or the graph. All they do is they throw in really big words to try to like mess you up, but just keep it like super simple. Okay. So when I look at this, it says nutrient agar plates. It says wild type E. coli. Wild type is essentially going to be what we see in nature, like what's in the wild. And then we have E. coli with some sort of plasmid in it. Okay, so if I go up to the reading, it tells me that the plasmid allows it to be antibiotic resistant. So if I keep looking, it tells me that if it's completely like grayed in, it means that the bacteria is like growing a ton. So what I do is every time I have a diagram or a picture, I always say to myself, what are they showing me? Okay, I kind of ignore everything else and I try to like simplify it for myself, because once I figure out what they're showing me, I can kind of figure out any question after that. Okay, so with no ampicillin, so with no medicine, the bacteria is grown like wild. Okay, then all of a sudden we add the antibiotic and they all die. Okay, cool. Well, now if I am going to mix up the E. coli with an antibiotic resistant plasmid, so now they're resistant to the antibiotic, I can now see that in this particular situation in like, I guess Q4, if you will, all of a sudden I'm having bacteria grow because now they're expressing those antibiotic resistant traits. So I could ask any question about this, but as long as you could just simplify that in your head, then you're like golden, like you can totally do it. Okay, so my suggestion for you is any FRQ, I want you guys to look at the graph and title, usually look at the captions and say, what are they showing me? Ignore the big words, right? No one cares it's E. coli. I'd be like, okay, it's just some bacteria. So that's an example of like a lab-based investigation. It's not going to be something like, what should you have done in your leaf chad lab? Like that's not the question that it's going to be. They would give you enough that if you didn't actually do the lab, you can still answer the question. All right. So the next FRQ question, number 4, is going to be some sort of phenomenon that we've talked about, and then a disruption. So maybe it's going to talk about the photosynthesis and respiration and what happens if we have a forest fire. So something dealing with a disruption, what would actually happen? Okay. The next 2 questions, question 5 is going to give you some sort of visual picture that you just have to kind of follow and then just answer questions. And then question 6 is going to be some sort of data analysis. Okay, so you have an idea of what you're going to be getting, you just don't know the topics, but you're going to be totally fine. Okay, so here are some tips and tricks. I want you to think about your AP test like an Easter egg hunt. Okay, do you guys remember when you were little and you were like hunting for Easter eggs and you were all like dressed up really cute? And if you had siblings, you guys are like totally matched. Well, and you had to go find Easter eggs and you had a cute little basket and you went around grabbing all the Easter eggs. It didn't matter the order that you got those eggs. It didn't matter, right? It didn't matter the location. It didn't matter the sequence that you got it. All that mattered was at the end, you had this huge basket with these eggs like overflowing. That is what you should think about your AP Bio test, okay? For your multiple choice and your FRQ. It doesn't matter where you're getting the points. Your job is to get as many as you can. In order to pass the test, you really need to just get 51% of the exam right. So that's like a little more than half of the multiple choice, and then a little more than half of the FRQ, and voila, you have totally passed your AP Bio exam. We really want you to get the 5 though. So when you're approaching the problems, remember that there's no penalty for a wrong answer. So when you're getting that multiple choice and you're going for it, y'all, when in doubt, just choose an answer. Don't leave it blank, okay? In terms of your FRQ, like I said before, you want to get all the Easter eggs that you can. It doesn't matter the order that you got them. All that order matters is going to be the total that you got them at the end, okay? So the other thing I want you to know is that every graph or model that they give you is going to relate to one of the questions, okay? So make sure that you guys are looking at the models, looking at the graphs, and you'll be able to answer most of them. I want you to remember the KISS method, which is to keep it simple, silly, okay? Ignore the big words. They're going to give you the biggest words in the entire world. But if you see topoisomerase, you know it is an enzyme because it ends in ase, okay? So if they give you the blah, blah, blah protein, the cytochrome C protein, whatever, it is a protein. What are the basic things we know about proteins, okay? The other advice I want to give you is ATP. Yes, it's adenosine triphosphate, but it also means to only answer the prompt. Don't go more or less. If they say to identify 2 things and move on, okay? Your test is going to be the application of what you've learned, not so much recall. So you don't have to memorize dates or people or anything like that. It's going to be how can you apply what you learned, okay? So every single question that we do is publicly released from College Board. So if at any point you're like, I really feel like I need to go back to that, you can access the presentation. There's like a little green button underneath that you can just click on it and the slides will pop up, and also the recording will as well, okay? All right, so task verbs. It is important that you know what these actually mean, right? So that way you can answer the question. So describe is what is happening, right? So like what actually is, like what do you see is going on? So I see the graph is, you know, slowly increasing, and then it reaches a peak at, you know, 20 degrees Celsius. Like what is happening? Explain means the why. Like why do you think that actually happened? Identify our 1 or 2 word explanations, and that is it. Do not put it in a complete sentence. Justify means to prove it. If you're saying this, tell me the why, okay? Analyze means look at the graph and then come up with some sort of like conclusion, like why, like what's something that we can say about this particular situation. And then prediction, we kind of all know what a prediction is. All right, so we're gonna go right into science practice 4. Okay, y'all, get ready. So, Alexandra, you are not screwed. That's why you're here with us, friend. We got you. All right. So, science practice 4 is about graphing. Okay. So, what are the things that we need to know about graphing? If you were ever asked to make a graph, I want you to make sure that you have a title. Your title should include your independent and dependent variable. Your title should be clear enough that when I read it, it gives us an idea as to what you're graphing. And that's why you want to include your IV and your DV. Make sure your variables are labeled. What is on your X? What is your Y? Make sure you have clear plotting, okay? You can only use a blue or black pen, so you can't really like use different colors for legends or things like that, but you can do strikes or dots or things like that. And then you want to plot accurately. You want to plot error bars if they give you error bars. And then you want to make sure that you choose the right graph. Okay, so all those things will give you multiple points on the actual FRQ, which is nice. All right, so here is an example of FRQ. Okay, it is usually really, really long. This would be an example of what your second FRQ would look like, where you have some sort of data table and then you have some sort of graphing to go with it and some questions. But I want you, again, to just look essentially at what they gave you, okay? So we're going to go ahead and do little parts of it together. Okay, so let's read. I'm trying to make it bigger. Whoops. Sorry, guys. Okay, so let's go ahead and read the FRQ. Okay, you can read it to yourself, but I will also read it out loud. So it says, Table 1, the effect of caffeine on memory in bees. So we have 2 treatments. We have the control group and we have caffeine. We have memory measured at 10 minutes, and then measured at 24 hours. So in flowering plants, pollination is a process that leads to the fertilization of an egg and the production of seeds. Some flowers attract pollinators, such as bees, using visual and chemical cues. When a bee visits a flower, in addition to transferring pollen, the bee can take nectar from the flower and use it to make honey for the colony. Nectar contains sugar, but certain plants also produce caffeine in the nectar. Caffeine is a bitter tasting compound that can be toxic to insects at high concentrations. To investigate the role of caffeine in nectar, a group of researchers studied the effect of caffeine on bee behavior. The results of an experiment to test the effect of caffeine on bees' memory on a nectar are found in Table 1. Okay, I'm going to set a timer for about 6 minutes. And I want you guys to do question A on your own sheet of paper, okay? So on your own sheet of paper, I want you to draw me a graph of that data, okay? So I'm going to go ahead and I'm going to start the timer. And again, figure out what type of graph you're going to show me. What do the units look like? What goes on the X? What goes on the Y? Yes, you do want to plot those error bars, okay? So if you don't remember how to plot the error bars, you essentially go up and down that amount. So like for 10 minutes for the control, you would plot 0.72 and then go up 0.09 and then down 0.09. And that's how you draw your error bars. Okay, so let's go ahead and I'm going to start the timer and I want you guys to go ahead and graph and label everything that you got. All right, my loves. So time is up. Okay. I am reading your chats and I want you guys to take a deep breath. Okay. Every teacher that you had did the best that they could. And so the reason that you're with us in crayons is to get caught up. All right. So we got you. Big deep breath. Okay. I want you to focus on the task at hand. And that's the only way that you're actually going to actually learn something. Okay. So the question that I saw a lot essentially was, how do I know what type of graph to draw? Okay, so whenever you are comparing data, so like in this particular situation, we have a control group, and then we have a group that we gave caffeine to, and then we're measuring the memory in those two groups. So whenever there's a comparison, that's going to be a bar graph. Okay. Whenever you have some sort of change over time, then that's going to be a line graph. If they ever ask for a correlation, that's going to be a scatter plot. And those are the three main ones you need to know. Yes, I know there's like a pie chart. I've yet to see anyone have to make a pie chart, any of these things. So if I were to write something down on my notes, if you don't know what graphs to draw, I would say if it's comparing data sets, bar graph. If it's change over time, line graph. If it's asking for a correlation, that's a scatter plot. Okay, those are the three main ones. So again, line graph is change over time. That's what I'm going to write down. Then I'd write down comparison is a bar graph. And if I see the keyword correlation, I know it's going to be some sort of scatter plot. Okay. Now you might see different types of graphs, but those are the only three that I've ever seen you guys have to actually create on the AP Bio exam itself, okay? And so in terms of what we're looking at here, I saw a lot of questions about the standard error of the mean and whether you had to like multiply it by 2, because in AP Bio, we do use 2 standard error of the mean. They know for College Board that your time is a value. So I have not seen them ever ask you to calculate 2 standard of the mean. It's usually just provided. So if you guys look on the graph itself, they gave you 2 standard error of the mean. So you didn't have to multiply by 2 or anything like that. If they gave you just standard of the mean, then yes, you'd multiply it by 2. But that is such a small like point that I would just move on with that. Okay. So if you guys look, what you do is, let me show you the type of graph that you should have gotten. Oh, Lordy Lou. There we go. Okay. So let's see how you guys did. So I'm going to move myself off the screen so hopefully it becomes a little bigger for you guys. So essentially for your title, you would have the effect of caffeine on memory of bees. So we have our independent, we have our dependent variable. We have the actual treatment groups on our x-axis. So we have the caffeine group, and then we're going to have the control, and then we have the short term and the long term. And then we have the actual memory. And so what you can see is in the data table, it gave you the average, right? So like for the control for 10 minutes was 0.72. So you created whatever scale you want to do, you make your 0.72, and then you're going to go up and down whatever they give you. So it was 0.72 plus or minus 0.09. So that means go up 0.09 and then also go down 0.09. That's what that means, okay? In terms of the scale, it can literally be anything that you want. I just saw that. It's whatever you want it to be, okay? As long as it fits in the graph, you're good. You can even like add extra lines if you're like, I need to go above and beyond it. So again, when you guys look at the data table that they give you, I want you to, the music's going to start again, you guys. But essentially, if you read it, like 0.83, that's the average. Then you're going to go up 0.07 and then also down 0.07. And that's kind of where those lines are coming from. Okay, so let's see what you guys got on the graph. So this was a 3-point question. If you made it a bar graph, you got a point. If you have your X and Y axes correct and you have a title, that's another point. And then if you have error bars on there, that's your 3rd point. Okay, so did anybody get 3 out of 3? Can you give yourself a little shout out? 3 out of 3? It's okay if you switched out like the caffeine with the control. That's fine. No big deal. Did anybody get 3 out of 3 points? 3 out of 3. Very good. There it is. Good job, you guys. Great job. Beautiful. Now, say you forgot to put the error bars on there. you would still get 2 out of 3. So it's not like, it's not all or nothing. Okay. So they want to give you as many points as possible. And if you forget something, then you won't get credit for that. But again, it's the Easter egg hunt, right? Like you want to get as many Easter eggs as you can. So maybe you forgot to put a title, or it wasn't a clear enough title, that you won't get points for that, but you don't get deducted points either. Okay. So yes, every graph you do, make sure you have a title, make sure you have your X and Y axes labeled, make sure you have your units on there, and then those are nice, kind of easy points. Okay, so let's try another part of it. Okay, so now let's look at the next question. Okay, so looking at your graph. So looking at your graph, I want you to describe the effect of caffeine on the short-term memory and the long-term memory. So look at your graphs, and underneath your graph on your own sheet of paper, let's give you about 1 minute. I want you to tell me, what effect does caffeine have on short-term and long-term? Okay, so take a minute and see if you can write out. It can be just 2 sentences. So what effect does caffeine have on short-term memory? And then your second sentence will be the effect of caffeine on long-term memory. Okay, I'll give you about 1 minute to try to write it down. All right, so let's see how we did. Okay, so whenever there are error bars involved in the actual graph itself, and then asks you to form some sort of conclusion, it's usually looking for significance. So a lot of you guys use statistical significance or difference in your response. Perfect. Okay, so if I were to look on the short-term memory, we would have little to no difference. Okay, so when I compared to the control for short term, I recognize that my error bars are kind of overlapping. Okay, so the error bars are indicating some data above and below the average. So essentially, if your error bars are overlapping, then there's really no significance between the two. So some of the data that you got in the control, you also got in your experiment. So there's really no difference. So you would say something along the lines of, caffeine had little to no effect in the bees' memory on short-term memory because the error bars are overlapping. Boom, period, full credit. Okay. But now if I go long-term, we can see that the error bars are not overlapping at all, right? Like Lordy Lou, like down here, the control has this data, and then long-term over here is this data. There is no overlapping. So we would say caffeine does have an effect on the long-term memory of bees because the error bars are not overlapping. Or if you want to talk about significance, you would say that there's more of a significant difference in the data or in the memory of bees long-term wise. And that would be 2 points right there. Perfect. Okay. Did anybody get 2 out of 2? Give yourself a shout out. Anybody get 2 out of 2? I know you guys did. 2 out of 2. Yay! Good job, you guys. Don't you wish this was like the real test? Because then we could all pass and move on. Perfect. All right. So I know that was a little intense. So I'm going to ask you the next question that my daughter actually wants to ask you, my 9-year-old. You ready for her question? So Miss Madison, if you can help me put it in the poll. My daughter, Cecilia, said this is a real question. She wants to know, do big kids peel their string cheese, or do they just bite it? Let me know. What do big kids do? That's what she wants to know. All right, big kids, what do you do? Are you a peeler of the string cheese, or do you just chomp it? Now it makes me want to have some string cheese. I guess this will tell me a little bit about your personality as well. Okay, so most of us are peelits. All right, chompets. There's about 7 of you guys out there. How do you do that and not feel like, oh, I gotta peel it? All right, awesome. Thank you for sharing. I will let my little know that the big kids like to peel their string cheese and not just chomp it. All right, awesome. Thank you guys for sharing. All right, so now let's move on to science practice 5. Okay, so first science practice is graphing. Okay, every time you're asked to make a graph, give a title, give your axes, graph accurately. You only have to plot the error bars if they give it to you. They don't always give you the error bars, okay? So that's not always going to be a thing. If you see error bars, make sure you talk about significance in the actual response, right? So in my notes, what I would write down and maybe star is that if the error bars are crossing over, then there is no significance between the data because the error bars are overlapping. If the error bars are not overlapping, then there is some significant difference between the two. Okay, so that's kind of what they're looking for. Science practice 5 is doing a little more with the graphing, except this time it's focusing a little bit more on the calculations and experimental designs. We're going to kind of go right into that. Okay, so first thing I want you to know is the difference between the independent and dependent variable. Like, yes, you should know the definitions. The independent variable is kind of what you're testing, and the dependent is how you're going to measure what you're testing. But in terms of science practice 5, it's really focusing on, do you know where to put the variables on the graph? And so there's different ways of remembering how to do this, but I've always used this one little trick, and so I want to share it with you. So if I look at the XY axes, on the left, okay, so on the Y, it looks like the back part of the D for dependent variable. Like I can literally draw the axes and then I'd be like, oh, that's a D. So dependent variable goes on this part. And then on the bottom where the x-axis is, that's like the top part of the capital letter I. Okay, so from this, I would literally just draw this down, and I go, oh, my independent variable is always going to be on this x-axis, and my dependent's always going to be on the y. And that's just a little trick to kind of help you guys out with the differences between the two. Okay, because sometimes what they'll do is they will give you a graph, and then like a simple question will be, what's the dependent variable? And it's like, just look at the axes labels; it'll be there for you. Okay. The other 2 words that students kind of get confused is the difference between controls and then constants. So your control group is always going to be used for comparison with your experimental group, right? So our control is, we're going to say, oh, here's the control, and then the effect of different amounts of water, right? And we can kind of compare. So just think control and think comparison. In terms of constants, those are going to be used for validity of the experiment. So we know every experiment can only have one thing that's kind of different, which would be the independent variable. So if you're going to measure the amount of water in plants, those plants better have the same amount of soil, they better be in the same amount of light, they better be in the same location, same humidity, right? Those are all your constants. So if they ask you on an FRQ to design an experiment, make sure you talk about the independent, dependent variable. What are your control? What's your control group? What's your constants going to be? And then you want to talk about having a large sample size. Like always just write in there a large sample size or repeated trials. Those 2 kind of keywords always give you like points on the FRQ because every experiment, you have to have a large sample size. Okay. Again, you want to make sure that you're going to use the right type of graph. We just talked about this. And then some words that get kind of tricky. So I would write these down if you guys don't know these definitions, because every now and then, of course, College Board has to be a little tricky. Okay. So we all know that a hypothesis is an educated guess, right? We were taught that in 7th grade. We know that it usually is going to be in an if-then format. So if you're asked experimental design, you always kind of put in what your hypothesis is. But every now and then, they want to throw in a null hypothesis and then an alternative hypothesis. When I think null, I think no, okay? So the null hypothesis is always going to say that there's no difference between the variables. There's no relationship, okay? So various amounts of water will not impact plant growth. That would be the null hypothesis, that there is no relationship. So in my notes, I would write null hypothesis equals no relationship. So just always say does not have an effect of, or there is no relationship between this and this. That's what it's looking for. So make sure you use the word no, okay? Alternative hypothesis is kind of what you're used to saying. So like if I were to say, if I increase the amount of water, then plant growth will also increase. That would be what's called an alternative hypothesis, saying that there is a connection between the two. Positive control versus negative control, okay? So a positive control is when you're trying to test the validity of your equipment, okay? So if I am going to measure, say, like the amount of fat in a solution, then I'm going to take just pure fat, I'm going to add my drops to it, and it's going to turn positive because I know it's fat. So whenever you're testing, is what you have actually working? That's called a positive control, right? Maybe I want to transform bacteria, like in an FRQ, the question that we looked at where we did bacterial transformation with the plasmids. I want to make sure that I'm using the right bacteria. So I would probably swab a regular Petri dish, and then I'd be like, oh, the bacteria is on there. And I know that I'm experimenting with the right bacteria. So your positive control is looking for your validity. A negative control is usually the control that you're used to. It's like the group that nothing is done to, okay? So like if I'm measuring the amounts of water in plants, your control group would be the one with no water, right? Like we can predict what's going to happen to it. But that's the difference between the two. So positive control is more like the validity of a reagent or a bacteria. And when you think negative control, that's just the normal thing I think about when I think control, okay? They just add these fancy words. So alternative hypothesis, you could almost cross off alternative, like hypothesis is just the normal thing that you're used to. So those are just kind of key words to think about. So if on an FRQ, you were asked to design an experiment, make sure you put a hypothesis, your independent variable, your dependent variable, large population size, repeated trials, your constants and controls. And that's what they're looking for. Do not waste your time doing step-by-steps. They are not asking you to retell them the steps of the Leaf Child Lab, right? They're not like step one is, that's not what they're looking for. They're looking for big pictures, okay? And then you're gonna move on from that. Something that you're gonna see a lot on the exam is your rate of change. You should know the formula for rate of change, okay? So essentially it's the slope, which is rise over run. You're gonna take two data points, and you're going to essentially do y2 minus y1 over x2 over x1. So we essentially are looking for the slope, okay? So the steeper the slope, the faster the rate is gonna be. If the line's going up, we know that the rate is increasing. If the line's going down, then we know the rate is decreasing. And what they'll do, and you can see right here, this was a multiple choice question that you guys did a couple nights ago in one of the crams. And they wanted to know the rate, and they'll always give you two data points, okay? And so what you do is you say, okay, they want to know the rate between this point and this point. And so I'm going to go ahead and I'm going to do the differences in the y, and then divide it by the differences in the x, and then go ahead and give yourself some sort of unit, okay? So that's what they're looking for in the rate. So you might see the rate as a multiple choice question, or you might see it as an FRQ, but you will always be given a chart and or a data table, and then you have to figure out how to do that, okay? So I'm gonna give you 2 minutes, and I can't wait to hear what this music is. And in the 2 minutes, I want you, on your piece of paper with your calculator, I want you to calculate the rate of change of bacterial growth between 5 and 10 minutes, okay? So I'm gonna go ahead and move myself out of the screen so it can be a little bigger. All right, you guys did a great job. All right, so let's check it out. Oh, Lordy Lou. There we go. All right, so essentially, we have rise over 1, right? We just talked about the different times of minutes. So 200 minus 80 over 10 minus 5 gives you 24. I apologize. I forgot to put the units on the first graph. So I'm sorry. I know that you guys all did it right. Beautiful. Okay. So you're going to see a lot of rate of change. So be ready for that. Okay. All right. So now my son has a question for you. He wants to know, would you rather have the superpower of flight, strength, or invisibility? Which one? What would the big kids rather like? Would you rather be able to fly, be the strongest person in the world, or be invisible? Oh, it's kind of a tie. No one's saying strength? Okay, there's one strength. Fly like an All right, that's interesting. Okay, I'll let him know that most of you guys wanted, it was a tie, I would say, between flight and invisibility. Oh, Tara, not in here, you don't, my love. Interesting. All right, beautiful. Okay. All right, my dears, before we move on, okay, we need to take a 5-minute break because we are about to hit Chi-Square, Hardy-Weinberg, the really, really hard stuff, okay? So I want you to take a 5-minute break. I want you to go to the bathroom, grab a snack, grab some water, and then we're going to come back together and then be ready to get the hard math stuff, okay? So far we've done experimental design. We've done rate of change. We've gone over the formatting, the FRQs, and basic graphing. And so when we come back, we're going to be doing the very hard stuff, okay? So about 5 minutes. All right, my loves, here we go. So I saw that a lot of you guys had questions when we were doing the error bars about what like a line graph would look like with error bars. And so I was going to go right into chi-square, but I thought let's like wait for the heavy hitter at the very end, right before we do our Samasoa song that everyone's talking about. So let me show you an FRQ of a line graph with an error bar, and that way you can kind of see the difference between the two. Okay, so let me go ahead and hit next slide, obviously. The best thing to do when it comes to your FRQs is going to be looking at a graph. What can you tell me about what's going on based off what you see? So before we look at the actual graph, I have like two pictures to show you real quick. Okay, so here are two pictures. Based off the only thing that I've given you, which are the two pictures, what is a conclusion that you could make about one of these pictures based off what you see? And then what's the evidence for that? So if you're going to say, yeah, the polar bear is struggling, tell me why. Why is the polar bear struggling, right? So tell me something about one of the pictures and then also tell me the why behind it. Either one, pick one. Everyone's choosing the polar bear. What about this cute little squirrel right here? What's going on here? Beautiful. There's competition for the same food source between squirrels and the birds. Habitat loss, competition. Habitats being destroyed. Survival of the fittest. Good. Very good. Oh, look at you guys using those evolution terms. Very nice. Miss Kari would be proud of you guys. Good. Okay. So you can look at a picture and then you can tell me what you see and then you can support it. And that's exactly what you guys did. Okay. So the squirrel is competing for food because they have the same food source. The polar bear's habitat is diminishing because the temperature is increasing. Okay, so what you're actually doing is a method called CER, which is claim, evidence, and reasoning. Okay, so I want you to remember this method and apply it to every single graph or model that you're going to get. All they're asking you to do in almost every single graph that they give you is what do you see? And then how do you support it? And then what do you think it means? Okay, so don't be intimidated by their graphs and images. Just take a deep breath and say, okay, let me think about the squirrels. Like, what am I seeing? Like, no one is expecting you to be an expert at any of these things. They just want you to go, what are you seeing? Support what you're seeing, what you just said, and then why do you think you're seeing what you're doing? That's every FRQ, and you can do that, okay? So here's an example of a beautiful line graph that has the error bars. Okay, so I'm going to kind of move into the wing, so hopefully the screen will get bigger for you. Okay, so here is a beautiful graph for the FRQ. And the first thing you're going to do when you look at a graph is I want you to look at the title. Okay, you're not going to freak out. You're going to take a deep breath and say, what is it showing me? So it says in figure 1, it's the effect of KAR. Who cares that we don't know what that is? No one cares. It's KAR, whatever. And TMB. So these two chemicals on seed germination in these specific plants. And there's some error bars. Beautiful. Okay, so in my head, I'm going to use my KISS method and I'm going to say, okay, what are they actually showing me? So I got time on the X, I have seed germination on the Y, and they're testing the effects of these particular chemicals on the germination of seeds. Okay. So when looking at this graph, what is my dependent variable? What did they physically measure? What's my dependent variable? Remember what we learned about the I and the D? Which one's my dependent variable? Is it time or is it seed germination? Beautiful. It's seed germination, right? So remember what we did where we have on the Y, that's like the backbone of the D, and then the bottom is the I. Okay, so the independent variable is the time, and then the seed germination is your dependent. So they measured seed germination, and you have the effect of these various different chemicals. So the first line is showing just KAR on seed germination. The second one showing the control group. Then you have both the treatments together, and then you have just TMB. Remember, you do not have to be an expert in these chemicals. You will have never have seen any of these words. It does not matter. In my head, I go, KAR is a chemical, TMB is a chemical. Great, I can read that and figure it out, okay? So what you're gonna do, if they said to you, given this graph, I want you to form some sort of conclusion. There are multiple conclusions. Just like you guys did in your pictures with the squirrel and the bears, you guys all came up with different conclusions, and then you kind of supported it with why you thought that. This is the same scenario. If they asked you to make a conclusion, just pick one. There's like 50 of them in here. Like literally just pick one. And what you would do is you would compare it to the control, because we know the control is always something that you're going to compare it with too. So don't get intimidated by these big graphs. No one's asking you to summarize the entire thing. Summarize one part of it and then be able to justify it, okay? So as long as you can justify what you're saying, you're going to get the points, okay? So in terms of the evidence, what you do is whenever it says to justify your conclusion, it means to support what you said with the data. So if you said KAR increases the time of seed germination in comparison to control, the evidence is the data. Always use the data, right? So if you said the bear is fighting for his life, and then you told me because he's on that little piece of ice, it's the same concept. KAR increases the time of seed germination. It has a time of 12 minutes in comparison to the control that has like 14. I can't really see it that far from here, but that's what you're going to say is you're going to justify means use the actual numbers, like use the numbers. That's what they're looking for. Literally use the numbers. Okay. And then the reasoning is going to be where you bring in the biology. Like, you know, maybe for seed germination, this chemical provides the most nutrients, that provides the most habitable situation for these plants to germinate. Okay. So even though you guys might not know what germination means, essentially you can just kind of like wing it with the graph that itself. Okay. Does anybody know what germination means? Can you guys put it? What is germination? That's kind of a science word. What is germination? The seed growing, sprouting. Perfect. Okay. So you can have a seed that's not germinating, right? Like a sunflower seed, right? But the moment that you put that seed in its ideal condition, we say it is germinating. So germinating is essentially the sprouting of the seed. It has the right nutrients, and it also has the water, the sunlight. That's germination, okay? Growth of seeds. Beautiful. So now let's go ahead and do some like practice with these actual questions. So using this graph, the first FRQ question says, here are 4 claims and they want you to support their claims. So we're going to do 2 of them together, okay? Well, 2 of them at a time, I should say. So looking at the graph, remember, we don't care that we don't know the chemical makeup of this, whatever, doesn't matter, okay? Take a deep breath, and you have 2 claims. Your job is to support the claim of what they said. So the first claim says, KAR alone affects the timing of seed germination. So what would we say that would support what they just said? Okay, so if you said the squirrel is competing for food, the justification would be both the squirrel and the birds are both fighting, right? So if we're saying KAR alone affects the timing of the seed, you have to figure out what line are they referring to. So they're referring to the first one, right? Because the first line is KAR, the second's the control, the third is both of them, and the fourth is TMB. So essentially, they're trying to get you to zoom in on one of the lines. You have to kind of figure out which one you're going to be referring to. Now, what you want to do is you want to use actual data in your justification. So you don't want to just say that there is a steeper slope. Like, give me some actual numbers to go with it. Okay, so I would say If it's doing the timing, so KAR alone is going to germinate at about 12 minutes in comparison to the control that germinated at about, let's say, 14 minutes or 18. Okay, that's what you would say. So your justification, y'all need to remember to use actual data from the graph. Don't just talk about us. Like, I want you to use the actual data. Yes, the slope is a great thing to talk about, but to give those full points, they want you to use the actual numbers. Okay, so the actual time itself. Very good. All right, the next one says KAR alone affects the percentage of seeds that germinate. So now we're looking at percentage of seed germination. So let's go to KAR, and we would say KAR has about 95% germination rate in comparison to the control that has about 80%. Okay, so use the data, use the numbers, and compare it to the control. And that's how you would do those two. Okay, I'm going to give you guys about 1 minute. I want you to try these next two. Those are 2 more claims, and I want you to support that. Justify, prove that those claims are correct with data from the graph. Okay, so let's talk about it. Okay, so here are 2 claims, and then they want you to justify it, which basically means support it with information from the graph. So if we look, TMB alone affects the timing of seed germination. So we have to go, okay, where do we got to zone in? Oh, they just want TMB alone. And it talks about timing. So I need to talk about the timing while comparing it to the control. So TMB alone is going to germinate at about 22 minutes versus the control, which is about 18 minutes. Boom, done. That's it. Use the numbers. Number 4, TMB alone affects the percentage of seeds. So TMB had about 12% seed germination in comparison to the control that had about 70%. Okay, so they gave you the claim, and your job was to use the numbers to support what they said. Okay, you're going to get a lot of this in terms of your FRQs and in the multiple choice. They might give you a claim and say which one's going to justify the situation. So if you see justify, remember that simply means to prove what they just said. Okay, so I would write down in my notes, justify means to prove the claim using the numbers. So use the numbers. Okay, it might not be right, but you want to justify what they just said. And that's kind of the point of that. Okay, now a second part of this FRQ is another graph. Okay, it's really data table. But we're not going to freak out. Okay, we're going to just read the title, and we're going to kind of just use the KISS method to figure out what they're telling us. So it says the effect of chemical treatment and water rinsing on germination. So I have 6 treatment groups on the left-hand side. I have KAR and TMB. Again, I'm not freaking out that I don't know what those stand for. It don't matter. They're 2 different chemicals. I could have elemental P, okay? 2 different chemicals. And then water. And what I can see is an actual result. So I don't really have actual qualitative data. This time I have quantitative. Like, it tells me if I have something that's different or something from the control. The plus indicates that that was added, and the minus indicates nothing. So like if I look at treatment group 1, nothing is done to it. That's my control group. I get some sort of result. And then in treatment group 2, when I just add KAR, I get something different from the control. And then in 3, I add TMB, I get something different. Now for 4, I'm looking at the effect of rinsing, and I can tell that because that was in the title, the effect of water rinsing. So if I just rinse the seeds with water, I get some sort of result. If I have KAR and water, I get something different, and then so forth. So just look at the graph. Okay, so now let's answer some questions to go with it. All right, here we go. Here's the question. It says, make a claim about the effect of rinsing on the binding of KAR in the seed and about the effect of binding TMB to the receptor in the seed. So identify the appropriate treatment groups and results from the table that when compared with the controls provide support for the claim. Okay, so they're asking me for 2 things. Make a claim about the effect of rinsing on KAR. So I need to find where it's KAR and water. So if I look, the KAR and water, oh, that's treatment group 5. And then the other one they are referring to is just the effect of TMB, and that is it, not water. So the one with TMB that has a plus sign is group 3, okay? So all they want you to do now is to just form a claim and then indicate the result and what treatment group it came from, okay? So now we are zoning in on just those 2 parts. So your response could be something along the lines of, because you always want to compare it to the control, always compare it to the control. So you would say, when TMB in treatment group 3 produces a result that's different than the control, then I would say, when KAR is rinsed with water, as shown in treatment group 5, can you guys hear me now? Yes. Okay. All right. Yay. All right, now I gotta figure out how to fix my screen. Hold on, I'm getting there, you guys. All right, Jeffrey or Madison, can one of you guys try to screen share for me? Whoop. Okay, phew. Okay, we're good. Can you guys still hear me okay? Okay, thank goodness. All right, I apologize for that. Okay, so let's go back to that problem set. What I wanted you guys to recognize is that in everything that we just answered, did we have to actually know anything biology related? Like literally, like think about everything that you learned. Did we actually, in order to answer that entire FRQ, have to know anything biology? No. Doesn't that make you kind of feel better? That would be an example of like one of your long FRQs. So that should make you feel a bit better. The fact that like just from the diagram and the data table, you answered almost all the questions. Okay. So subtitles back. Okay. Let me put it on 1 second. Is that any better? There we go. Okay. So it should make you feel better. This idea that they're going to give you everything you need and you actually didn't have to have this like complex understanding of like photosynthesis or respiration. So even though it talked about plants, it didn't really, which is kind of nice. Okay, we were able to answer it. So this would be an example of like your first question, your long FRQ. Now, remember that time is valuable for your FRQs. So like one of the questions we didn't answer on here, it's kind of hard to see on your screen, but you did have to apply the biology and it asked like a little more complex about like a process. But if you're like, you know what, I'm going to move on to question 2. You can do that. Remember, it's your Easter egg hunt. You don't have to answer every question and don't get stuck. Like you could easily go, okay, they're giving me a claim. Justify just means I need to support it with the numbers. I can do that. Boom, boom, boom. I can look at the data table. Ooh, it's asking me something complex about like the Krebs cycle. Moving on from that, let me move on to the next one. That is totally allowed and that's totally okay. And that's actually encouraged. Do not get stuck on something you don't know. If you feel like you're like, I don't even know what that word is, let's just move on. And that's totally fine. Okay. Let's do another practice question. So here would be an example of a short FRQ. So this might be more of like one of the questions like 5 and 6, where they're going to give you some sort of data analysis and then ask you to form some sort of like conclusion of some kind. Okay. So essentially, here's a short FRQ. And here's kind of it zoomed in. So you can kind of get a better idea. So looking at the graph, I'll take myself into the wing so that it kind of appears a little bit bigger for you guys. We have a graph, okay? And it says figure 1 is the effect of exercise on blood prolactin levels in adult males. The data represents the means of 2 standards of the means, okay? So again, who cares that we don't know what prolactin is? It don't matter, right? So we have blood prolactin levels on our y-axis, and then we have with exercise and without on the x. And then we have some error bars, okay? So essentially, number 1 says, justify the use of without exercise treatment as a control. My hint for you is anytime and every time that it talks about justifying the use of a control, I want you to immediately go control comparison. It is used in order to compare the experimental data to. It could be this generic sentence, boom, done, period. You're going to get the point. Okay. So anything that talks about justifying that, make sure you say that. Number 2 says, using evidence from specific treatments, determine whether prolactin release changes after exercise. Justify your answer. Okay. So take about 1 minute. look at those error bars. We talked about those error bars. Is there significant release of prolactin after exercise? Okay, so take a minute and then let's talk about it. Perfect. You guys got it. Okay. The results are not significant, right? So those error bars are all crossing. So you would say, due to the error bars crossing, there's not significance or statistical significance between the release of prolactin with or without exercise. So we can't really tell. There really is no significance because all the numbers are basically the same, right? Like if we look, all of those error bars are overlapping. Okay. So anytime you see error bars overlapping and they ask you to justify your response, what they're looking for is something about significance. And then put in there error bars are overlapping or they're not. And that's what they are going for. Absolutely, Angelina. I apologize for that. Okay. Right. So now we have seen error bars. We have seen how many times you're going to be asked to make a claim and justify. So in my notes, I would have down a claim is basically a conclusion, and that as long as I can support it, it's going to be right. The other thing I would write down in my notes is going to be that justify means to use actual data points. Okay, that would be the other thing I would write down. Use the data points to support what I just said. The other thing that I write down is that if it talks about justifying and there's error bars, it's looking for significance, and you want to talk about those error bars crossing or not. And then the last thing I would have written down as my special notes is going to be essentially to just look at the graph and be able to answer the question. Okay, so here's another question for you. If this is my short FRQ, did I actually have to know anything biology content related? Did you have to know anything biology content related to answer that question? Nope. Again, okay? So big deep breaths. If you don't understand the Krebs cycle, Calvin cycle, big deep breaths, you can still do really, really well in the FRQ. We just did one long FRQ and a short FRQ, just looking at a graph, and we didn't have to input anything from our actual content or curriculum. Okay, so big deep breaths. You can do this. Okay. Now, I'm not saying that you don't have to know anything biology related. Most of the time you'll have like part A, B, C, and D, and like one of the parts will be something biology related. But if you're not, if you don't know like the process of the Krebs cycle and the Calvin cycle, just don't do that part. Because what I don't want you to do is to do a short FRQ and then you get to that part and you're like stuck and you're like, oh my gosh, you don't remember what that is. And now you've wasted 10 minutes because the next question will be the same thing where it'll be read a graph, read a graph, read a graph, justify claim. And then maybe the last part will be something content related, okay? Maybe you go back and do those. You don't want to miss the easy points. Again, it's an Easter egg hunt and you want to get all the points that you can. Don't get stuck on something. You can justify, you can claim, you can predict, you can make all those things just by looking at the graph for the model, okay? So you can do that nice and easy. Okay, we're going to take now a 3-minute break, you guys. We are exactly halfway done. Okay, the second half will be our heavy hitter, but we're halfway done. So take 3 minutes, have a moment, grab some water, have a stretch, and we're going to come back together. All right, my loves, here we go. You may not be ready for this exam, but this exam is happening and we're going to help you get ready. Okay. So remember, don't focus so much on how overwhelmed you are. Be present, be in the moment, take a deep breath, and let's keep going. We are halfway done. Okay. All right. So let's do another practice FRQ. So with this next one, you are being given some more graphs. Hopefully you're seeing this theme of like it's graph after graph after graph, but that's beautiful because everything's in the graph and it makes life like so much easier. Okay. So this is basically your entire science practices is analyzing the graphs. Okay. So let's go ahead and see what they gave us. So I'm going to go ahead and move myself out. So hopefully it makes the screen a little bit bigger for you guys. So let's see what this graph is. Okay. Again, big deep breaths, read the captions. Let's look at figure 1 first. So it says figure 1, the cell number compared with the number of cells at whatever measurement OU bang, whatever. Okay, so great. Normal human kidney cells or NHK. So these are kidney cells. And polycystic kidney disease, PKD, were treated with increasing concentrations of this particular chemical. And I see the error bar. So I go KISS method. What do I got? I got this particular treatment of OU-bang. Who cares that we don't know what this is? We have number of cells produced, and NHK stands for normal, healthy kidney cells, and PKD are ones that have some sort of kidney disease. Okay, got it. Figure 2 is the percent of sodium, potassium, ATPase activity of those same exact cells with increasing OU-bang. Okay. Again, don't get lost in the words. Just look at what they gave us. We're looking at our squirrel. We're looking at our bear. Big deep breath. And let's figure out what the question is. Now, the other thing they did was they gave us this other figure to look at. Okay. Now, when they give you figures like this, I want you to say, let me read the caption first. So it's a signal transduction pathway hypothesized to play a role in the increased number of those PKD cells. Whatever, great. Now, look at the direction of the arrows. That usually gives you context clues as to what's happening in this actual diagram. Okay, don't get lost in the big words, right? Just go, what am I looking at? Am I looking at the bear on the ice? Like, what do I see? I don't care what these big words are, okay? And then you'll be able to kind of figure out what's going on. So look at the direction of the arrows. Sometimes they even number step 1, step 2, step 3. So just look at what they gave you, okay? So starting at the top, I have some sort of enzyme. Does anybody know how I knew that was an enzyme? How do I know sodium, potassium, ATPase is an enzyme? What's the giveaway there? ACE. Beautiful. Okay. So in my head, I go, it's an enzyme. It does something with sodium, potassium, and it probably makes ATP if I had to guess, right? Most of the time, your biology words will tell you like exactly what's happening. So the chemical binds, and then MEC gets phosphorylated, and then that's then going to phosphorylate ERK. Again, who cares that we don't know what these are? And then a phosphorylated ERK then is going to go ahead and we're going to transcribe target genes. So this chemical binds to this enzyme, which then is going to phosphorylate or activate a whole bunch of other things. And then it's going to be able to go through the nuclear envelope into the DNA itself. I'm just following the arrows. And that's all that you have to do for most of those. So the first thing in my head, I go, oh, that's kind of like, I remember signal transduction pathways, right? It even says what it is. So we receive the information, we're going to relay it, and then something's going to happen. Okay, so now let's go ahead and answer the 2 questions, okay? Using this graph, and I'm going to give you some time to do this. Describe the characteristics of the cell membrane that prevent simple diffusion of sodium and potassium across the membrane. And then B, explain why ATP is required for the activity. So figure out what part of the figure they're showing you Keep it simple. And I'm going to go ahead and give you 2 minutes. Try to, you can write it on your own paper. You can put it in the chat. I'll leave it up to you. Okay. Answer the 2 questions using the figure, using your basic understanding of biology. Okay. About 2 minutes. Go for it. The blob that you see is labeled. Remember, look at what they gave you. You can do this. All right, my loves, let's see how we did. You guys did really good. So here is what the rubric would have said. Okay, so for the first question, it says to describe the characteristics of a cell membrane that would prevent simple diffusion. So you could have said any of the following. Any of the following would have given you the point. You could have talked about the fact that the cell membrane is semi-permeable. Okay, the idea that you have a hydrophilic head and then the tails are hydrophobic, which gives you that semi-permeable membrane so that it would prevent the simple diffusion of sodium and potassium. Okay, perfect. You guys did that beautifully. In terms of the second one, it says explain why the ATP is required for the activity of the sodium potassium pump. And it's exactly what you guys were talking about, was this idea of active transport and that you're going against the concentration gradient. Anytime ATP is required, that means you're going against the concentration gradient. So instead of going from areas of high concentration to low concentration, then you're just going the opposite. Okay, so very simplistic. You could have answered both of these with one sentence. Okay, you guys did really good with that one. Good job. All right, so my daughter has a question for you before we move on to the next complex part of this actual problem. She wants to know if you could have any pet in the world and it would not hurt you, what would you own? A snake. Snake, a lot of you guys would want a snake. A chipmunk, how cute would that be? I guess it would hurt you if you had one for you. A lion, cute. Oh, Rachel, a lemur, I love that. A shark would be nice. Very nice. All right. Thank you for sharing. I'll be sure to let her know what you guys said. Okay. I want to go back because some of you guys are asking some questions about the responses to this. And you want to keep everything nice and simple. Okay. So the first question, what I would do is I would say, okay, what's a characteristic of the cell membrane that prevents simple diffusion? And I would say, oh, things that are large or are unable to go through the nonpolar tails aren't able to move in. So it's something about being semi-permeable. The second one, it says, why is ATP required? And somebody had said, well, how do you know it's required? Well, it says it in the problem. It says that ATP is required. It's also in the name of the enzyme. So they're not going to trick you up like that, okay? So anytime ATP is required and it deals with movement across the membrane, always correlate it to active transport, okay? This idea that it's going to go from low concentration to high concentration. Somebody else was like, how do I know what these things are? It's just labeled. It's just labeled, okay? So the interesting thing about these two is that it's only asking you questions about the first part of the figure. Like it's not even asking you anything about the rest of them. And please do not say the blob in your response. Y'all better use the names ATP, or sodium, potassium, whatever the name is, just use that name. Okay, so here's the next question. It says, what is the dependent variable in figure 1? So first of all, find figure 1. Remember what I taught you about the I and the D. What is my dependent variable? What is it? What's a dependent variable in figure 1? Boom, number of cells. You would have gotten a point right then and there. Done. Perfect. Okay. Now let's go ahead and do the next two parts of this FRQ. So A says, justify the use of normal human kidney cells as a control in the experiments. Ooh, keyword control. My go-to response is going to be used as a comparison to the experimental groups. Boom, done. There's your point. Okay, so you kind of see these patterns. Okay, B says, justify the use of using a range of OU-bane concentrations in figure 1. Why do you think it is a good idea to use a range of concentrations versus just one concentration? Why do you guys think it's a good idea to use a whole range? Beautiful. Great job, you guys. To show the huge to show the essentially the optimal levels of OU-bane over the different concentrations, to have a larger population size, right, for validity of the experiment. You want to be able to look at how the range and the number of cells are correlated. More beautiful. You guys did great. Perfect. Great job, everybody. That would have been 2 points right then and there. Okay. Question C. Based on the data shown in figure 2, so we're going to zone in on figure 2, so the diagram on the right, describe the relationship between the concentration of OU-bane and the sodium potassium ATPase activity in both the NHK and PKD cells. Lordy Lou, sorry. So what is the relationship that you guys see? What do you see in the graph? Describe what you see. Are we seeing the bear on the iceberg? Just tell me what you see. Who cares that we don't know what these words are? What relationship do we see in figure 2? Great job, you guys. You got it. It's so funky for no reason. Please don't put that on the actual exam. Funny though. Very good. Okay, so we would say as the concentration of OUmin increases, the percent of sodium, potassium, HbS activity decreases. Boom, done. You got your actual point and you move on. Okay, so you guys have answered it in one sentence and it's exactly what it should be. Okay, great job. Okay, so when you guys look at that entire FRQ, like of all the parts that you did, I hope you recognize that it was only the first two parts that really asked you questions about the cell membrane, and all the other parts were just reading the graph. Okay, so I want to try to get you to relax. I want you to take, this would be a long FRQ. Okay, this is a long FRQ. This would be like one of the first ones where you have a bunch of data and you have to try to answer it. So this would be a long one. Okay, so for this one, I want you guys to take a deep breath and recognize that you can answer these questions. Maybe you can't answer every part of it, but you're going to be able to do most of it, which is phenomenal. So even if you didn't know anything about the cell membrane, you can still get a whole bunch of points. Again, you would have gotten a whole bunch of Easter eggs, right, without having to answer anything that was biology related. Okay. All right. So my son has a question for you. What pets do you guys have? What pets do you have? I have a leopard gecko, which was inherited from one of my former AP Bio students. And I also have a pit mastiff. Her name is Bella. She's as cute as can be. A bearded dragon. I so want one of those. Oh, cute. A pet cow. No way. I would love that. Have you ever met a 6 and 9-year-old? They got lots of questions. Another leopard gecko person. A chameleon. So fun. A little brother named Matthew. Don't tell him that. 30 horses. Oh my goodness gracious. Lordy Lou. That's a lot. A bunny, a brother. You guys, be nice to your siblings. Oh my gosh, Jay is bringing it back. All right, we'll move forward so y'all can hear that stinking song. I know you love it. Okay, so go time. All right, formula sheet. You guys, make sure your calculator's ready, okay? And you've had a good stretch. Formula sheet. You will be given a formula sheet. You don't have to bring one your own. My suggestion would be to just Google formula sheet. If maybe one of my student hosts can go ahead and also put that in the chat for them. But essentially, you can look at the formula sheet in advance and just go, do I know how to use this? Do I know what to do? Okay, and that's how I would use this. So you will maybe have to do a little statistical analysis. We know that that's one of those science practices that you have to do. If it talks about probability, thank you guys, you're going to be going ahead and you're going to just look at what they gave you. Okay, so everything you need is on the formula sheet. You just have to be able to use it. Okay, so let's talk about chi-square. Big deep breaths. I'm going to use the KISS method and break it down for you. Okay, no, it isn't a downfall. We are hoping for a chi-square problem. We got this. Okay, so let's talk about chi-square. So let me put my face back in here because I get really excited about chi-square. This is what you're going to, don't say heck no, we got this. Remember, we're going to start where we are and we're going to lock in and we're going to go ahead and just do this. Okay, so I'm going to try to simplify it as much as I can. Okay, I call it schematifying it, like just bringing it down. My AP stat people, I need you to bring it down and like keep it super simple with us, okay? So chi-square is a statistical test that allows us to determine if what we observed is what we expected. So I want you to imagine that I gave you, each of you, and I wish I could, a little M&M bag. And then I said, I want you to count how many different colors are in your M&Ms. If you look at the Mars website, the candy website, they actually have their goal of what their percentages should be. So I think it's something like 20% of every bag should be blue, 30% should be brown, 10% should be green, right? That's what they think it's supposed to be. But if I open up my M&M bag and I'm supposed to have 10 blue and I have 2, in my head, I would go, I got cheated, right? Something happened. And so the chi-square allows you to determine if what you see in your bag is actually significant to what we were supposed to get. And that's what the chi-square test does. It allows you to determine if there's a significant difference or not. If I ran a genetics test and I was supposed to get 75 brown bunnies and I got 76, is that really significant? Probably not. But the chi-square allows you to mathematically determine if there's a significance or not. And so you have the formula here, you have the variables, and you have the chi-square table. What I want you to know for the chi-square table, this is the table you're going to get, is I want you to always use the p-value of 0.05. Don't ask me why. It doesn't matter. Just know in your head, always use 0.05. That is it. Don't even look at the other one. Okay, 0.05. And what you're going to do to figure out the value in the chart, which is called your critical value, is you have to determine your degrees of freedom. Degrees of freedom is the categories you have minus 1. So in my notes, what I'm writing down, I'm going to write down that I'm always going to use a p-value of 0.05, and I'm going to star that. And then I'm going to write down that degrees of freedom is the amount of categories I have minus 1. Okay, so if I have 5 different color of M&Ms, my degrees of freedom would be 5 minus 1, which would be 4. Okay, and where those two numbers intersect, that is called your critical value. So this is a critical value chart. It is the highest acceptable number. So that's the next thing I write down in my notes. The critical value is the highest acceptable number. Okay, so let's go ahead and do a practice with this. So here are some of those key terms, but it kind of makes more sense if we just kind of like work our way through it. So essentially, let's do a problem. All right, y'all, here we go. In cats, long hair is recessive to short hair. You observe 42 long hair babies and 8 short hair babies, test the hypothesis that both parents are heterozygous, and run a chi-square analysis. So you are basically saying that both parents are heterozygous and somebody mixed it up and you want to make sure that you actually got the right result. So you're going to run this analysis that's going to tell you whether or not they're both heterozygotes or not. Okay, so that's essentially what we're doing. Like, did I mess up, or is what I saw what I actually got? Okay, so this is going to be your chi-square table. Okay, and so they are going to ask you for something called the null hypothesis. And we talked about the null hypothesis already, that there's no correlation between the two. Okay, so when it comes to chi-square, your null is always going to be that there's no significant statistical difference between observed and the expected. There's going to be no difference. That's always the null. There's no significant statistical difference between observed and the expected. So what you do is literally plug and chug what they gave you. Okay, so big deep breaths. We can do this. So they told us that they physically counted 42 bunnies had long hair and 8 had short. Okay, they told us that. So all I did was I just put it in the data table. Your job is to figure out the expected. Well, if I have 2 heterozygote parents, I'm going to go ahead and create a Punnett square, 2 heterozygotes, so big L, little l, big L, little l, and I have that 3-to-1 ratio. So what I'm going to do is I'm going to say, okay, I have in total 50 bunnies. Okay, 50 bunnies. And I just figured out that 75% of the 50 is how many long I should have. So all you have to do is do 0.75 times 50. That gives you a number. In terms of rounding, I usually tell my students to go about 2 decimal places. That's kind of like my go-to unless they tell you otherwise. Like I know it sounds silly to have like 12.5 bunnies, but when it comes to a mathematical problem that would be in your FRQ, they're going to allow a range of data. So if you choose 2 decimal places or 1 decimal place, just be consistent with it, okay? So at the end, if you end up with like 14.3 and the answer was 14.5, you're still gonna get the credit, okay? As long as you're close enough, you're gonna be good. Okay. So now we are going to go ahead and put in the expected. Okay, so again, I just did the Punnett square and I figured out 75% of the population. So 75% of 50 gave me the 37.5. 25% of the 50 then gives me 12.5. And now you just plug and chug. Okay, so now you do O minus E squared over E. So I would do 42 minus 35.5 squared divided by E 37.5. And I put in that number. And then I would do the same thing for the next one, okay? It's just plug and chug, and this would be given to you. Your chi-square is the sum of the last column. Okay, again, your chi-square is the sum of the very last column. So all I did was add the numbers. So I end up with a chi-square of 2.16, whatever it is, 2.16. Okay, that's my chi-square number. Once you have the chi-square, then you have to figure out your critical value. Okay, so the chi-square is what you calculated. It was the sum of that last column. So critical value, here I go. I know it's 0.05. I remember Ms. Shoemate told me that my degrees of freedom is categories minus 1. So I had, if I go back into my category, I had long hair, short hair. So 2 minus 1 is going to be 1. So I need to figure out where my numbers are going to intersect. So with the p-value 0.05, and then I have the degrees of freedom of 1, my critical value is 3.84. Okay. Let's talk about what we did for chi-square. Number 1, the null is always, there's going to be no significant statistical difference between observed and expected. You can use that same sentence for every single problem they give you. There's no significant statistical difference between observed and expected. That's your null. Boom, done. Then you're going to fill in the table with what they physically tell you they observe. Then you're going to calculate the expected. Then you fill out that table. And then the chi-square is the sum of that information. Okay, so we summed it up and we ended up with 2.16. The second thing you do is figure out your critical value. Your critical value is in the chart. You find the p-value 0.05. Your degrees of freedom is going to be the categories minus 1. We had long hair, short hair, so 2 minus 1 is 1. Our critical value is 3.84. Okay, so this is how I want you to think about this. The critical value is the highest acceptable number. Okay, so these are the highest acceptable numbers that you can ever get. That's it. So the highest number we can get is 3.84, and we got 2. So what you do is now you have to form your conclusion. And that was the whole point of this, is to form a conclusion. Is what we expected what we observed, or is it not? Did something happen? Okay. So I want you to think about that critical value like a wall. Okay. It's the highest acceptable number. The critical value is a wall. And then think about it like a number line going from the left at 0 all the way to the bigger number on the right. Okay. Any number that is less than that wall is not significant. If I asked you to walk up to a wall, you could easily just walk up to a wall. Okay, like don't do what this person is doing, just like running through it. Okay, it's not significant. But if you had a number greater than this wall and I said to you, hey, I want you to climb over that wall, that's a significant amount of energy. Okay, so if the highest acceptable number is basically 3 and we got 2, we would say our results are not significant. And that would be the conclusion that you would say. So if your chi-square is less than the highest acceptable number, then what you do is we say that the results are not significant. Therefore, the null is accepted. Okay, so the null is accepted. when the results are not significant. So we're going to say there is no significant statistical difference between observed and expected. Therefore, our parents are probably heterozygous. Okay. If our chi-square was greater than that and our results were significant, then we say, you know what, we reject the null because the results were significant. So that means that our parents were probably not heterozygotes. Okay. And that's kind of how you remember that. So I want you to remember that the critical value is a wall. Anything less than that wall, the results are not significant, right? What you saw is what you observed. Okay, your Mars candy, even though you're supposed to have 20 M&Ms, you had 21. The results are not significant, so we accept it. But anything that's greater than that wall, that's significant. Climbing a wall is significant, and that's when you would reject it. Okay, so you kind of have to remember those two different conclusions, and then you're golden. Okay, so all you're gonna do is write in what they gave you. Determine your expected. Usually it's going to be a Punnett square of some kind, or they're going to tell you that it's going to be expected. Then you fill out the chart, find your chi-square, which is the sum. Then you find your degrees of freedom, which is categories -1, and then determine your conclusion, okay? If your chi-square is less than that critical value wall, then that big wall, remember, then we're going to go ahead. I know it's like field trip, I know, but we're going to just say, you can either say accept or reject in AP Bio, thank goodness. Okay. All right. Big deep breaths. So one of the ones that you probably had a bunch of questions about, and we're actually going to kind of try it so that you can kind of understand how you would apply chi-square to a scientific problem. So this would be a problem that you might have is like a long FRQ. You have a choice chamber, and let's look at what they gave us. We got glucose soaked cotton on one side, and we have dry cotton on the other side, and we're going to have some bugs. Okay. So the first question is, predict the distribution of flies in the chamber after 10 minutes. So if I were to drop flies into these chambers, predict where they're going to go and then justify your prediction. So we're just looking at the bear on the ice. Like, what do we see? And then what's your proof for what you see? So where are flies going to go? What's your prediction? Perfect. Okay. So the flies are going to be distributed on the side with the glucose soaked cotton because that is a food source that they would be attracted to. Boom, you just got 2 points. Easy. Okay. Done. 2 points. Nice and easy. All right. Number 2, propose one specific improvement to each of the following parts of the experimental design and explain how the modification would affect the experiment. So what's something, if this was the only experiment you had, like this is it. Okay, look at the picture. What is something we can do to improve the control? And then how do you think that would actually improve the experiment? And then what is something that we could probably do better on? That's an environmental factor. And then how do you think that would improve the experiment? Very good. You guys are doing great with that. Okay, so in terms of the control, you would want to soak the dry cotton in water because you're kind of testing multiple things, right? Like maybe they like the moisture or the dryness, or is it the food that they're attracted to? It's kind of a hard tell, right? So you would go ahead and probably soak the dry cotton in some sort of water, right? So because that way you're taking away, is it the texture of it? Okay, that's perfect. In terms of environmental factors, okay, so you could say things like different concentrations of glucose, okay? You could say, there's a multitude of things you could say. You could talk about the duration of the experiment, right? You could talk about the time of day, the amount of light, different temperatures. You could alter the humidity, the wind, right? Like all those things. You just have to pick one thing, and then you could say that it could attribute to the movement of the flies. Like as long as it's like pretty practical, then they're just going to give it to you, which is kind of nice. Okay. So here's the second part. So that's kind of like the experimental design, which is one of those science practices. So here's the chi-square part, and this is what it would look like. So here we have the distribution of flies at 1 minute, 10 minutes, and it says perform a chi-square for the 10-minute data. So it only wants you to do it for the 10-minute data, not the 1 minute. It's not even looking for that. So just for the 10-minute data, okay? Specify the null and then enter the values into a chart, and they give you an actual chart. So here's a chart that you would have, okay? And this one we're just gonna kind of walk through because of timing. I wanna make sure I get to Hardy-Weinberg. So here's the chart. The null is always that there's going to be no significant statistical difference between observed and the expected. That's the null. That's your statement. Use it over and over and over. There's no significant statistical difference between the observed and the expected. Then we're going to go ahead and put in what they gave us from the data table, right? Remember, they gave us a number 45, 3, and 12. We just plugged it in, 45, 3, and 12. Now, for the expected, if the null is always saying that there's going to be really no difference, then essentially it should be this like even distribution of flies. So if I have 60 flies all together, then, and if I'm saying there's no difference, then I would just go 20, 20, 20. Okay. What's the even number of it? Then we're going to plug and chug. We end up with a chi-square of 48.9. Okay, that's a big number. It doesn't matter what it is. It's chi-square equals 48.9, which is just the sum of that last column. Then I know that I'm going to go to my p-value 0.05. And I know that I have 3 categories. I have the end of a banana, the middle, and then the unripe banana. So I have 3 categories. So then I know that 3 minus 1 is 2. So my highest acceptable number is 5.99. Okay. The highest acceptable number is basically 6. And I got 48.29. What does that mean? Are my results significant? If the highest acceptable number is 5.99 and I got 48, are my results significant? Yes, super significant. That's right. Holy moly. It is super significant. The highest acceptable number is 6. I got 48.9. Lordy Lou, right? So essentially that means because my chi-square is greater than that critical value, I am rejecting the null because there is a significant statistical difference between observed and the expected. So what I expected to see and what I thought I got is like totally different. Perfect. Okay, great job. All right, we are now going to go into everyone's awesome. Lordy Lou, I know, I'm sorry, that's my statement. The next one we're going to go into is Hardy-Weinberg. All right, y'all, we got this. Okay, Hardy-Weinberg. Big deep breaths. Okay, we're going to like simplify this. Okay, I know that's why we're going over these. I know it's your least 2 favorite. All right, Hardy-Weinberg. Let's just humidify this and make this super simple. Okay. So Hardy-Weinberg are 2 scientists that wanted to be able to predict the changes in allele frequencies in a population. Okay, so in a population, you have a bunch of species that are living together and they interact with each other daily, they interbreed, they produce fertile offspring. Okay, And so they want to pool all their genes together. So like if I were to take in that population, so all the dogs in my neighborhood, and I wanted to look at a specific gene in all of them, I'm going to look at all those letters. I'm going to pull them together. That's called a gene pool. And so Hardy-Weinberg basically created an equation that allows you to look at those allele frequencies and how they change over time. And if we were to do that, and we recognize that the allele frequencies are staying the same, we say the population's at equilibrium, okay? So when dealing with Hardy-Weinberg, if a population's frequencies are staying the same, we say that it is in equilibrium. And when that is happening, then we have these 5 conditions that are being met. We don't have any mutations happening. We have random mating, natural selection, a very large population size, and then no gene flow, okay? But if any of those things are not met, then the population is going to be evolving. So there are 2 equations that you need to know, okay? So I simplify this really, really, really easily, but you have to write this down in order to understand how to do this, okay? My math people, you probably just get this and can move on, but everyone else, I want you to actually write this down, okay? On your paper, like on your actual paper, I want you to write the equation. So everyone write it with me on your paper. P plus Q equals 1. Okay, so this, you're literally writing P plus Q equals 1. Okay, P stands for the frequency of the dominant allele. So let's just give it the big letter A. Okay, so underneath, I want you to put a little arrow and say the frequency of the dominant allele, big A. It's also in the slide. And then for Q, that is the frequency of the recessive allele, little a. So I always just label it. This is your key equation. I want you to know that your job is to look at any problem they give you and try to figure out P and Q, okay? The only time that they will be giving you P and Q is if they say those exact same words. The frequency of the dominant allele, then they're giving you P. The frequency of the recessive allele, they're giving you Q. Now, if they're talking about genotypes, then this is what they are referring to, okay? Now, this is the second equation, but a lot of students get confused because they're always like, oh, I'll just set it to 1 and then I'll move on. But this is only true if the population is at equilibrium. So most of the time it doesn't equal 1, okay? This is the equation I want you to remember. If I give you P, you find Q. It always equals 1. So if P equals 0.5, Q is 0.5. If I give you Q is 0.8, P is 0.2. You just put the numbers together, okay? P squared, I want you to think about it as a variable. P squared is big A, big A. 2PQ is going to be big A, little a. And Q squared equals little a, little a. Think about them as independent variables, okay? So this is kind of how I like essentially remember it all. So P is the frequency of the dominant allele, big A. Q is the frequency of the recessive allele. Frequency means decimal. So anytime you put a number into these charts, I call these my variable charts, it has to be a decimal. Has to be decimal, no whole numbers, okay? P squared is going to be big A, 2PQ is big A, little a, and Q squared is homozygous recessive. Okay, I want you to do a problem. So on your paper, I want you to draw me those 2 variable charts. Okay, so literally draw a box and put P and Q. Okay, so like literally, this is old school, I'm holding up to the screen. Draw a box, put P and Q. Then draw the other box and then put P squared, 2PQ, Q squared. Okay, don't make fun of my handwriting. So literally draw those 2 boxes. Okay. Your job is to figure out what are they giving me, and then how do I solve for P and Q? Because then after that, it's like totally plug and chug. Okay, so it says the scarlet tiger moth has the following genotypes. Calculate the allele and genotype frequencies. Okay, cool. So they're giving you immediately these 3 genotypes. So we can just plug those in. So remember, it has to be a decimal. So we're going to convert it to a decimal. Okay, so they told us 540 out of 640 moths is big A, big A. and then it gave us 70 out of 640 and so forth. So now we just convert them into a decimal, put in the calculator, okay? Now I can solve for P and Q. So Q squared equals 0.05. So my math people, all I have to do is isolate Q. So take the square root of both sides, that gives you Q. And then once I know Q, I have P, right? Because P plus Q equals 1. Right? So 0.78 plus 0.22, and then you're good to go. Okay? Now, let's try another one. Okay, so this one says, in a population of beetles, 60% have the homozygous recessive allele for colors. So homozygous recessive is Q squared. Okay, so they're giving me Q squared. Remember, it has to be a decimal, can't be a whole number. So that would be 0.6. Now I know Q squared, square root both sides to get Q. Subtract that number from 1, that gives you P. And then you're going to go ahead and do P squared and 2PQ. Okay, so, all right, my chat people, make sure you're focused and you're present, my loves. Okay, so let's focus on Hardy-Weinberg. So for Hardy-Weinberg, I want you to just always draw your variables tables. Remember that you want to use frequencies. Figure out what they gave you, then answer the question. Okay, so for this one, once my variable tables are filled in, the question is, what percent of the population are heterozygous? Well, heterozygous was 2PQ. So then I would say, oh, it's 35%. Okay, so you have to be able to go back and forth between percent and then from decimals. All right, so we'll make sure to do more Hardy-Weinberg tomorrow so that we can get through the rest of our equations. Okay, so we will do more of these tomorrow, I promise. Okay, so the next topic I want to go over with you guys is gene linkage and recombination frequency. Heterozygous, it would be heterozygous is 2PQ. Yeah, not the dominant one. Gene linkage basically means that the genes are linked together and then they get inherited together. So they basically talked about sex-linked traits, right? So the X chromosome has traits that are linked to the X chromosome, and then you have some traits that are linked to the Y chromosome, and then they're going to get passed on together. So linked genes are genes that get passed on together. And so we can do something that's called a recombination frequency, where we can calculate the odds if traits are being passed on together or not. So what you need to write down in your notes is that any recombination frequency, once you calculate it, is less than 50. So any number less than 50, we say that they are linked. They're linked traits, okay? So in order to figure out how to actually calculate it, the formula is the number of recombinants. Recombinant just means a different variation of mom and dad. That's what that means. Divided by the total offspring times 100. Okay, so again, total, so you have number of recombinants, so offspring that are not like mom and dad, divided by total offspring times 100. And that's going to give you a recombination frequency. Those frequencies equals 1 map unit apart. So if this is a chromosome right here, and this is a chromosome for fruit flies, recombinants are just offspring that look different than mom and dad. It's just a thing combination. They're a combination of mom and dad. That's all that it is. Okay. So if we were to look at this chromosome map, I've got all these different traits on this. Okay. And then I have different recombination frequencies. They're just the map units apart. So if I were to calculate the frequency between gene B and VG, I'm going to calculate it's 17%. That just means that there's 17 map units apart. I just need you to know that 1 recombination frequency equals 1 map unit. It's just kind of like if we were to put these genes in order. Okay, so let's do a problem. Okay, so let's say that you have a scientist that crosses a homozygous recessive purple-eyed vestigial winged fruit fly. Does anybody remember what vestigial means? My evolution people. What does vestigial mean? What's vestigial? Yeah, great question. What is vestigial? You guys tell me. What's vestigial mean? I saw it. Basically useless. Exactly. It's something that we no longer use. Okay. It's there, but we don't use it at all. Very good. Okay. So that's vestigial. So we have these flies that have purple eyes and wings they can't use. So let's say that that represents mama fly. And dada fly is a normal eyed fly. So that is going to be, let's say it's like a red fly, whatever it is, and normal wings. So the daddy has wings that we can't use. And so they make a cross and it tells you the percentage of 1,000 flies. Okay. And so what you do, and this is very simple, is you take the bigger numbers because most of the offspring are going to look just like mom and dad. So it says 42% basically look just like mom, and then 46 look just like dad. So you take the bigger numbers and we call those the parental offspring because they look like mom and dad. The recombinants are combinations of mom and dad. They do not look like mom and dad. They're totally different. Okay. So those are your recombinants because they don't look like mom and dad. So all you have to do is now use this formula. The number of recombinants, so 60 plus 60 is 120, divided by the total offspring times 100. So it's 120 divided by total offspring, which is 1,000 times 100. So I have a recombination frequency of 12%, which just means if I were to plot it on a chromosome map, they'd be about 12 map unit distance apart, which will make sense in a second. And the point of this is to say if those traits are linked or not. And the number is less than 50, therefore they're linked. Therefore, that basically means that the ability to have a color of eye and wings or no wings are going to get passed on together. They're linked genes. That's what that means. Okay, that was the whole point of doing that. Now, let's say you have a chart and they give you a whole bunch. I'll go back. Sorry. Because it says 6% of 1,000, which is 60. So 6% looked like a different combination of mom, 6% looked like a different combination of dad, and that's where the 60 came from. Because 6% of the total population, they told us, was 1,000. Okay, so let's say I have this map, and I have all these recombination frequencies, and your job is to simply tell me the order of the genes on the chromosome. I want you to think about the chromosome like a number line, okay? And your job, correct, if their frequency is less than 50, they are linked, correct. That's what you need to remember. If you calculate all that and it's less than 50, you say the genes are linked. You're correct, okay? So what you're gonna do is you're gonna look at these recombination frequencies and say, okay, this is how you do it. Find the bigger number. And those two are going to be the farthest apart. Okay, so the bigger number is gene J and M. These are arbitrary genes. Okay, so I'm going to go ahead and put J and M on there. All right, and then I go, okay, those are 12 map distances apart, right? These are arbitrary units. Okay, and then I go, okay, now look at you. Very good. Y'all figured this out already. Now you go, okay, where does K and J fall within this map unit distances? So you kind of go, okay, between K and J is about 3 units, K and L is about 8. So you kind of think of it almost like a number line, and it's just like a puzzle, okay? So it's just a puzzle of what you got to do. So between L and M was 7, and between the K and L is 8, and then K and J, okay. So the order is going to be K, J, L, M. And it's just the order. You do not have to make this map like this at all. It's literally like just one of those logic puzzles where you go, okay, what's the order? If you don't understand this, take a deep breath. Okay, this is probably less than 1% of your AP test. Almost every question that we have done together tonight, remember that it's just been looking at a graph. These are just getting into the specifics. I just want to make sure you guys kind of have a rough idea as to what we're looking at. So the closer that they are, we kind of talk about them being inherited together, right? Like if genes are really, really far apart, then they're probably not going to be inherited together. But they kind of come as like a package deal. So like someone that has red hair usually has fair skin, because those traits are really close together and they get inherited together. That's kind of how we connect gene mapping and then the frequencies as well. All right, cell size. Okay, so for cell size, remember that you're going to be given a formula, surface area volume ratio, and that the point of talking about cell size is to relate it to diffusion. So I want you to remember that cells want to have a very high surface area to volume ratio. Blippy! They don't want to be too big because it'll take too long for things to diffuse through them, but they can't be too small because it has to hold all those organelles. So there's kind of like this sweet spot between what they want to be. So what they can do, and what you would do, is measure the surface area volume ratio. Again, the formula is on the formula sheet. You just kind of plug and chug it. And then you analyze what it is. What they want is they want to have a high surface area to volume ratio. And so cells are very good at kind of increasing their folding so that they're able to increase surface area without increasing their volume. That's kind of the point of that. All right, Simpson index. Okay, Simpson index. All right, here we go. Simpson index equation is basically an equation that deals with diversity. Okay, so you want to have a high diversity in a community so that it is going to survive any sort of disruption that comes in. If you have a community that has a lower diversity, then they're more susceptible to being extinct, right? If I have a higher diversity, there's a chance of communities being able to come back. Okay, so this is the formula. Like, Lordy Lou, look at that. It looks a little wild, but it's not that wild. Okay, so the Simpson Diversity Index, again, is just a formula that's going to help you calculate diversity. It's in the name, and Simpson's the guy that created it. Okay, or girl, I'm not really sure who Simpson was, but it's just a formula that's going to help us figure out if our community is going to be diverse or not. Okay, that's all that it is. So with that being said, let's do a little practice problem. Okay, I have 3 species and I have 2 communities, and I want you to calculate the Simpson Diversity Index and tell me which community is more diverse, which would be a healthier ecosystem because it could easily bounce back from any sort of disruption. Okay, so here's community 1. So you would calculate the Simpson Index for each of the communities. Okay, please use Lordy Lou. I would love that to go all over the place. So essentially, community 1, you're going to do 1 minus and then like a big bracket. You're going to do each of the population size divided by the total squared and then add them together. Okay, so that big E is going to basically be, Kathleen, that made me laugh. You got this, okay? That big E and that right, 1 minus E, that just means the summation, okay? So look how I did community 1. So 1 minus in the bracket. Look at community 1, 325 divided by the total 1,000 squared, plus 305 divided by 1,000 squared, plus 370 divided by 1,000 squared. You calculate it and you get 0.664. Okay, I want to give you a few minutes, and now I want you to do the same thing, but I want you to calculate the index for community 2. So figure out what I did for community 1 and go ahead and calculate the index for community 2, and let's see how you do. So again, I want you to figure out the index for community 2. Look at what I did for 1 and use the different numbers. And Elise, you're learning it now, baby. You got this. You guys did it. Great job. Okay, beautiful. So again, you guys are so smart. I'm very proud of you guys. Okay. So if we look, again, it's 1 minus in the brackets, each individual number divided by the total squared, and each of those are going to be added up. So if I had 4 species, you would do this for each of them. So now we have 2 numbers. And so we want to go ahead and form a conclusion. Okay. So we would then say that community 1 is going to have a higher diversity because we just calculated it. It literally, like, legit has the higher number. That's it. Okay. So community 1, we would say, has greater diversity, therefore greater resilience, maintains ecosystem stability. If a big disruption came in, then it would be able to bounce back quicker. Okay. And that's the point of Simpson's diversity index. Being able to calculate which community has a higher diversity and therefore be more resilient to disruptions. You guys, guess what time it is? It's the time everyone's been waiting for. Before, yeah, lots of math unlocked, I know. Before we do our quiz, so you guys can hear the song that you've been dying to hear. Madison, Jeffrey, Joyce, you guys have any final words of wisdom for our amazing individuals before I move on to the quiz?
Yeah. Well, you just sat there for around 2 hours and 30 minutes. So yeah, I mean, if you have any questions, ask it, but now it's time to demonstrate your knowledge. So yeah.
Madison, do you have any final words?
Similar to similar to Jeffrey, you guys just went through like a long time getting a bunch of information in your head, and those practices were probably very difficult, especially during the evening. So I'm very proud of you, and us as Fiveable are very proud. And yeah, similar to what Jeffrey said, we're gonna go do the quizzes you guys have been waiting for. So yeah.
All right, thank you guys. Joyce, do you have anything you want to add to it? All right, you guys, she's speechless. We did so much. All right, so we just completed the 4 nights, and we are going to go. You guys are making me laugh. Samasoa, I can't even say that word. So essentially, you guys have the grand finale tomorrow. Big deep breaths. We're gonna do more Hardy Weinberg tomorrow. Okay. And so please, I'm hoping that you're going to be able to join us for a final night of crams. It's going to be amazing. We're all going to be in this together. If you are on the East Coast, we're going to be going 7 to 10 p.m. And then luckily our exam is going to be Friday at noon for everybody. Okay. So at this point, I'm going to go ahead and start the quiz. Yes, I'll put on Samasoa. Hopefully it's going to work. Okay. And we're going to do some just practice multiple choice questions. They're not AP style, but they are enough that's going to kind of pull that content for you because we just did a lot of heavy math. As always, make sure you're going to follow us at Think Fiveable on social. And then I'm going to go ahead and let's do this. If you do not want to do the practical multiple choice and you guys want to take a nice rest, feel free to log off. Thank you for joining us. We appreciate you. I know that you want to hear the song though. And if you have any questions that you want to go ahead and put it in the actual chat, our student host will be there to help you. Samasoa. Okay. All right. So again, essentially my student hosts are there to answer any questions you guys have left. Feel free to log off, or let me go ahead and let me get started with the quiz. All right. Give me 1 second. All right, I just sent the link, and or if you want to do the QR code. Wait, can you guys hear the song? Can you hear it? How come I can't hear the song now? No, I don't hear the song. You guys, why isn't it playing? All right, hold on, give me a second. Oh, thank you. You guys, I don't hear the song. Why isn't it showing up? How do I unmute the web page? You guys, how do I share the audio? Now I want to hear the song.
I could try sharing my screen if that's possible.
Do you have the music, Jeffrey?
I mean, I can just toy around with this stuff.
Can you just play the music for them so they can at least hear the music?
All right, I'll share my screen. Actually, never mind.
No, Jeffrey, that's perfect. I can hear it. Can you guys hear it now? Jeffrey, they're calling you the king. Beautiful. All right, now let's answer the questions. Oh, it stops it. I didn't even know that. Good job, you guys. Jeffrey, they don't hear the word. Oh, there it is. Good job, everybody. are proteins. They're polypeptides. That was kind of a hard one, I know. So it says the polymers of proteins are polypeptides. If it was the monomers, it would be amino acids. Very good. Osmosis. Okay, so movement of water deals with osmosis. Good job. Jeffrey, got to play it again. Everyone wants to hear it. All right, competitive inhibitors versus non-competitive. So remember that competitive means it's going to compete with the inhibitor, like it physically competes on that active site. And then non-competitive means it's going to bind to that allosteric site and kind of change the shape of it. Interphase, good job. That happens right before mitosis. Very good. So centrodogma is a fancy way of saying protein synthesis. DNA to RNA to protein. All the above. Great job, you guys. The music stopped. It's gotta keep going. Jeffrey, we need you to play the music again. Producers, right? They have the highest amount of energy, and then it's going to go, and so that it will go down and down, down to the very top that has the least amount of energy. There it is. Okay, so I want you to look at the context clues, okay? So gravitropism, tropism is a plant's response to something. And in this case, the prefix is gravitropism. So it's a response to gravity. So that means it's going to either be C or D. And then negative means away and positive means towards. And so you might have learned this in your ethology unit when you talked about behaviors. So just remember positive means towards it, right? And then negative means away. So it's tropism is movement. Movement in relation to gravity, so away from gravity. Obviously, good job. Very good. It would then be a decline in every trophic level above it. Good job. All right, when you hear this song one more time. Keep on holding strong, y'all. About 4 more minutes. Let's get through as many as we can. Let's get a good review before we go to bed. They are the very first. Think about the word pioneer. Pioneer usually means like first to arrive. All right, you guys, last time you're going to hear your song before we go to bed. You can do this. Grasses, you guys all got that right. Look how smart you guys are. Good job. It is the first, the fourth, you guys. You count from the bottom up. So you have your first, which is going to be your producers, and then your primary consumers, secondary, and so forth. Very good, you guys. I am so proud of you. Every single person got that question right, okay? You guys are doing amazing. You know a lot more than you think you do. I am so proud of you guys. You held strong, okay? Tomorrow, the Samasura song is done. We will play it again tomorrow, but tomorrow's our grand finale. We're going to do more practice questions. We're going to give you tips and tricks again. We're going to do a little more math problems. We're going to kind of review everything one more time together. Lots of application of the information, okay? We appreciate you guys. You can do this. Big, deep breaths, okay? And you are going to be amazing. Please make sure to take our survey. Jeffrey had just put it into the chat for you guys. And we hope you have a great night. We'll be here for the next couple minutes to answer some questions. Thank you, guys. Have a good night. We'll see you